Welcome back. In today's video, I'll be talking about the cytokinesis process, which is nothing but the cell division after completion of replication and segregation of the chromosome and their movement towards the two poles of the newly formed cells. Now, there is an important process or important step, that is a cell will be divided with the help of septum formation. Okay, it means there will be a elongated cell with two chromosomes, but the cells needs to be divided. For that, the septum formation will be started from here, but this whole term is known as, or whole process basically is known as, divisome. Now, what is the first step of divisome? In case of the first step of divisome formation, the first one is, in case of E. coli, that is the FtsZ protein, which is nothing but the E. coli protein acts like a tubulin in case of that bacteria, that will be concentrated at the center of the cell to make a Z ring. Okay, Z stands for the Z ring. So FtsZ is the E. coli protein. It's a tubulin which will form a Z ring at the center of the cell. This is the first step of divisome formation, or the first step of cytokinesis in case of E. coli. Now, the major thing is, when the Z ring forms, it will not touches the membrane of the cell. Okay, for their anchoring to the cell membrane, there are two proteins which will help them into this. One is FtsA and another is ZA protein. These two proteins help to anchor this Z ring to the cell membrane of the elongated cell of E. coli. Now, another most important thing, that this Z ring always form at the center of the cell, not at the poles of the cell. Why? Because another protein complex which is known as MinCDE, this is also a protein belong in E. coli. This type of protein, this MinCDE, is basically concentrated at the poles. Okay, these proteins concentrated at the pole of the elongated cell. Whenever the MinC concentration is very high, this FtsZ concentration will be less. Whenever it's high, it's gonna be less. So MinC concentration is high at the pole of the elongated cell, so automatically that will make hindrance to concentrate more FtsZ protein at the pole, and due to that, that FtsZ protein will accumulate at the center of the cell. As MinC protein concentration is very less at the center, that is why Z ring always will be formed at the center of the cell due to this protein. Okay, so in this way, after Z ring formation, it will attach to the cell membrane, and the cell wall synthesis machinery will also attach to this, and new cell wall will be formed in between this, and that will give rise to progeny bacterial cells. In case of E. coli, this complete process, which I have already mentioned in my previous videos, reproductive stages, and then replication, then divisome formation, cytokinesis, this whole process will take about 20 minutes. In other bacteria, it may take about 60 minutes. So it's very less time. In case of E. coli, usually this whole process, starting from the cell elongation and up to the progeny, bacterial cell formation. It's a very fast process because E. coli can first, at a very high rate, they grow faster in a medium. So in this way, E. coli that will follow their cell cycle, replicate their chromosome, and then segregate their chromosome into the daughter cells, and will give rise progeny bacterial cells. Thank you so much for watching this video.